you going to be up? Are you going to be up? Are you going to be up by one? I have no idea if it's
We'll be in the New York Times. I haven't read it yet.
Still a little bit.
nature, there'll be some variation.
Good morning, everybody. Thank you. So we're here today on welcoming Diane Wang to campus as one of our interviewees for our positions in digital agriculture. We've been interviewing a number of different candidates over the last few weeks, focusing either on plant scale modeling, digital agriculture, up through sort of landscape scale modeling in digital agriculture. Diane did her PhD at Cornell with Susan McCooch, and we were just chatting a little bit about Susan and what things were like at Cornell this morning. Talk a little bit about Camp Lee.
That's good.
That could be bad.
No, it was she spent a little bit of time with a short postdoc at Cornell, and then now she's coming at the University of Buffalo. And she's going to be talking to us today about modeling the interplay of nature and nurture. It's a really beautiful title, actually. I appreciated reading your abstract and the thought that you put into your title. So Diane will be presenting here for the next hour or so. We'll transition into some faculty time to talk to her about her potential research program. So there's an opportunity to engage either here or afterwards. So we just want to say thank you, and we're looking forward to spending the next couple of days with you.
Great. Thank you so much, Mitch. Okay, well, good morning, everyone, and thank you so much for this opportunity to come and visit Purdue. It's really a pleasure to be here, my first time, and the rain has stopped, so it's a good day. So I'm a process-based modeler, and I'm currently at University at Buffalo working with an eco-hydrologist who's trained originally as an engineer, so it's been a really interesting period for me as we try to bridge fields between genetics and physiology and this sort of larger scale modeling. And it's a really exciting time for me, really interesting and exciting time to be trying to launch a career in agricultural plant sciences during this digital revolution that is really blurring the lines between the biological, physical, and digital spheres. So my work sits mostly in the realm of analytics. If we think about the digital framework, we can break it up into three main components: analytics, so this is like the data and the modeling; technological innovation, so these are more like the sensors that are getting the data; and communication systems that are really merging all these different things together. So the sensors are being able to send data to the cloud, which are being able to inform the models that we develop. So like I said before, I'm a process-based modeler, but I came, sorry about that. That's you. So I address mostly questions related to how different genotypes respond to different environments, and this is really what I think of as this interplay between nature and nurture. Right, so I think Mitch mentioned that I did my PhD originally in plant breeding and genetics at Cornell University, which is a little bit of a different path to what I'm doing right now. So there I had some really nice opportunities to participate in many different projects, and the themes of my research really revolved around two main components, and those were exploring natural genetic variation in cultivated Asian rice and their wild and needy relatives, as well as building resources for the global rice community. So these were mostly genomic tools and resources. So part of that was developing an imputation server to allow genomic imputation so that researchers around the world could try and integrate very disparate genetic resources, because as technologies develop, we're genotyping more and more on different platforms, on different germplasm panels, and these are very hard to integrate. And so imputation is one way to be able to add value to existing data sets, as well as to be able to merge them so that researchers can start to think about, okay, what is the best sort of subset that I want to use for my experiments later on. And so, but some of the challenges or the obstacles to this is high computational demand and lack of a reference panel. So we address these challenges in this project so researchers to be able to upload into a web-based application and get imputed data back at 5.2 million SNPs. But the focus of my dissertation work was really on trying to map out a blueprint of the genetic architecture of stem non-structural carbohydrates in rice. And so what these are is, so rice, like other cereals, maize, sugarcane, wheat, they accumulate non-structural carbohydrates in their stems. Like this, so what you see is you get an accumulation up until heading, after which point you see a decrease during grain filling, when a lot of this sort of storage NSCs are being remobilized to the grain. And this is thought to be potentially a good buffer for grain filling, especially under conditions with suboptimal environmental conditions for photosynthesis. So you can think of like drought or maybe lack of solar radiation. So it's kind of like putting money away to draw out later. But this trait is dynamic, so you can see it changes over time, and actually, interestingly enough, sometimes there's a little blip at the end of the season where some accessions will actually reaccumulate or start to accumulate again these NSCs. But as a grad student with limited resources, I could really only capture, if I wanted to scan across a large genetic pool, then I had to really be careful about what time points I could sample at. So really, I choose 2 main time points, heading and maturity. But really, I'm missing a lot of this dynamic, right? So we don't know what differences underlie any of these other components over developmental time across the succession. And so I liken it to sort of measuring how much water is in a bucket when someone's pouring water in and when there's holes leaking out. And this really highlights the challenge of working with dynamic traits. So here's a very simplistic example of human height. You can measure someone at 2 time points, let's say kindergarten and high school, but you really don't know sort of what path they took to get there. So this is totally unrealistic. Nobody grows like this, but it's just an example. Maybe some people are more linear, some people take a more sigmoidal shape, or this little step change right here. So this is sort of what I did. Had a fantastic growth spurt at 4th grade. My parents thought I was going to be a giant, and then it never really took off. So it's really this dimension of time. And at the end of my PhD, I struggled with this a lot. I knew I had the tools to be able to explore genetic diversity, but this dimension of time was really intractable to me. And I needed to be able to find a tool to be able to explore this idea of dynamic traits, because the idea of dynamic traits and time-varying environmental variables is so key, and I'm sure all the physiologists in this room will agree, to the processes that underlie G by E, or genotype by environment interaction. So this is what I decided to do a postdoc on, and now I'm in a geography department. Everyone is sort of confused about what I'm doing there, but this is what I'm doing there. Okay, so I'm doing biophysical process-based modeling, and so when I first started down on this journey, this is the definition I gave to my first talk about this new area. And it's really, at the time I said this was describing mechanisms, either known or hypothesized, that underlie physiological processes, turning them into mathematical equations, linking them together into a routine to simulate a time series of plant responses over time to different environmental scenarios. And these environmental scenarios also play out over time, so you can have things changing, all these different environmental variables changing. And I think that when I looked back on this a couple weeks ago, I thought, yeah, it still holds true for me today, but I just have really one addendum, and that is, here Modeling is not about math, it's not about equations at all. It's really, modeling is really an abstraction of reality. So we are surrounded by complexities in this world that's very hard to handle because they're all interacting, and modeling is a way to step back and be able to see the patterns in behavior and processes around us, and to be able to construct them in such a way that is useful to us. And so I think this is a really powerful tool. And if you're a visual learner like me, you can think of it this way. A process-based model has inputs and outputs. The inputs, we have system parameters that sort of describe the system. We have environmental scenarios, so like I said, the different variables that play out over time, and then they get ingested in a process-based model and then outputted traits, simulated traits over time. Okay, so you can think of them. Some of these parameters can describe behavior of growth, phenology and development, environmental variables, temperature, solar radiation, precipitation, vapor pressure deficit, wind speed, and the like. You can, depending on the model, I know many of you work with these types of models, they can be different. So for us, we have a very hydraulically sophisticated model that we work with, so we output things like leaf water potential or somal conductance, gas exchange traits, hydraulic conductivity. Okay, and we can also, so in this digital framework, we can think of a process-based model as an interface between the biological and physical world and then this digital world, which we're all trying to understand how that can be used for increasing efficiencies in agriculture. Spent the last 2 years really digging into the guts of this. So a little bit about the primary project that I'm on. This is an NSF plant genome funded project called Predicting Genotypic Variation in Growth and Yield Under Abiotic Stress through Biophysical Process Modeling. And it's a collaboration between several universities. We're at Buffalo. The main PI is out at Wyoming. Brent Ewers is a tree physiologist by training. UC Davis is doing a lot of the transcriptomic work. Dartmouth is providing the genotyping, and Wisconsin, they are developing the germplasm. And so one of the main goals of this project is to find the connections between omics data sets. So heterogeneous omics data sets, transcriptomics, metabolomics, these sorts of high dimensional data, and process-based modeling. And we use the model called TREES. It stands for Terrestrial Regional Ecosystem Exchange Simulator. And so for the rest of the talk, most of the time, like 99% of the time when I say trees, I mean the model and not trees, because that's a huge source of confusion when I start to discuss the model. And this has 4 primary modules, which are commonly found. Actually, 3 of them are commonly found in most crop models, and 1 is sort of unique to trees and this type of models used by the tree community. So it has carbon processes, nitrogen cycling, growth, development, and then hydraulics. In this proposal, we use Brassica rapa as a study species, and it has a couple of nice features. It's Brassica, you can think about utilizing some of the Arabidopsis resources, which I'm not super into, but that's one advantage. It has 3 main morphotypes, which are really interesting. So we have the oil seed, which is right over here. And this is primarily harvested for the seed to produce oil. We have the leafy types, so you can think of your Asian vegetables, your pak choi, napa cabbage, so these are the leafy types. And then you have your turnip forming types, which produce this really interesting below ground growth that's half root and half hypocotyl, which I'm sort of fascinated by. But then you can think about if we're going to have these really extreme morphotypes, every time you make a cross and develop population, you can have some very interesting combinations of traits. So we also want to use these process-based models specifically on different recombinant inbred lines developed from these intermorphotype crosses. So there's a couple of huge challenges to doing this. And the first is that the model that we use has been really well tested in woody perennial species like trees. And it doesn't take a brilliant person to know that this is not that, right? So when I joined, I thought, well, this isn't going to work for me. I don't really know, I don't know anything about process-based modeling, but this seems kind of strange. Like, we need to do something to the growth and development portion of the model, right? Clearly. So you can think of a long-lived perennial as having a very different life strategy to a short-lived crop plant. It's very fast-growing and undergoes these huge ontogenetic changes over a very short period of time, versus a tree who's kind of like, well, I've got many years to grow, this year's not so good, like I'll save my resources for next year, no problem. And so they're kind of more in flux with the environment, whereas with these short-lived plants, they're get in, get out, set seed, die, right? So we need to make this big overhaul. And the second is the extension of these models to population level, population interest-specific, to capture interest-specific variation. So for anyone who's used process-based models before, I don't know, has anyone? Yeah. Okay, so a big problem is parameterization, right? So like, how do you parameterize these models? There could be 100 parameters. And so if you're working on something like, oh, I study blue oak, or juniper, or pinyon pine, I can go and gather literature values, right? And that kind of works for the species. And that kind of works for things that are conserved across the species. But when we're talking about modeling interest-specific variation where you get really nuanced differences in parameters and their combinations, these don't exist in the literature. Nobody collects these and then publishes them. And so we need to find ways to be able to parameterize at scale and different ways to do so. So that's really what I'll talk about now. So this is